Hello, I'm Nicholas, all the way from the National University of Singapore, and I'm here to share with you work on warming waters and its effect on tropical sea jellies. So here we go. Singapore, if you don't know where it is, it lies on the southern tip of peninsular Malaysia. It's a low-lying island surrounded by the sea. So any effects of climate change, we feel it acutely. For instance, when the water warms out, the coral reefs around Singapore bleach, and most of the corals just give up and die. Singapore, when we did our marine research, historically we have been focused on coral reefs, but in recent years we're beginning to get more interested in sea jellies because there's increased sightings of them and increased incidence of them. Taxonomic records show that there are at least 40 species documented from our waters, but they're still poorly understood, not just in terms of their taxonomy, but also in terms of the biology. So if you're going to think about effects of climate change and on jellyfish around Singapore, how can we deal with it? We need to know about their biology. So we start off small, we start off modest. We want to find out how does warming waters affect tropical sea jellies. You hear narratives of this, warming oceans, there'll be jellyfish blooms, but you also know that we don't really know much in the tropics. Apart from that, warming sea temperatures not just affects the jellies directly, it also can affect them indirectly in terms of shifting food webs, right? So here we also wanted to test whether food availability, would that affect them. So we assessed them on two parts, on survivorship and the growth of polyps. Now before I continue this, what I'm sharing today is actually a minor part of a bigger study. So here we go, right? So how we went about doing it, we collected larvae from wild populations of Cassiopeia, like Seraphim yesterday. We cultured them, used them for our experiments in 3 different temperature treatments and 2 feeding regimes. One batch we fed it twice a week, and the other we overfeed it by 6 times a week. We conducted this experiment for over 28 days. We conducted it twice, and we looked at 3 different life stages of the Cassiopeia polyps, the 3 general ones. So we're still analyzing the data, but preliminary visualization, at least in terms of survivorship, we see that when they're really exposed to really stressful temperatures like 30 degrees and 32 degrees, they don't do well. The population crashes, and that's only when food is a limiting factor. But if you give them more food, they tend to, for those that are exposed to more stressful temperatures, they tend to do well. Maybe think of it like when you're stressed, you binge eat, you want to snack a lot. So that helps you tie over, those stressful times. And but you can only binge eat so much. If the stress continues, you die, right? Sort of situation. Looking at the growth patterns, where the red line indicates food limitation and the blue line indicates that you feed it with a lot of food. At higher temperatures, if food is a limiting factor, your growth is hindered. But if you start feeding it, you'll grow up fine. So in summary, just like Seraphim's excellent presentation yesterday with Cassiopeia, we concur with the view that they don't do well in higher temperatures, right? And it seems like for this one species, the growth, the survivorship is hindered at higher stressful temperatures. And food is a factor that we must consider, especially in our waters, Southeast Asian waters, where there's a growing industry of aquaculture in the seas. Now, like I said, this is part of a bigger work, right? We wanted to work on Chrysora chinensis, but when the money came, we couldn't find any individual, so we gave up on that. We wanted to look at gene expression, but we had issues getting the RNA from the polyps. Nonetheless, when life gives you lemons, you make lemonades. So this was published for the mitogenomics. Another part of the study looks at the pulsation rate of medusa. We compared 2 populations from the Red Sea and Singapore, and data on that is still being analyzed. I hope to share that with you soon. And with that, I thank the funders, the National Research Foundation from Singapore. This project was a collaboration between the Hebrew University of Jerusalem and the National University of Singapore. I would like to thank my colleagues from the Sea Aquarium sitting right there. They are the ones that helped me to keep the jellies alive. I'm terrible at that. And folks from Haifa University, who helped out a lot. With that, thank you.